This nerve impulse transmission story deals with the topic of what happens when an impulse gets to the end of an axon and has to be transmitted to the next cell. This is always an axon. I should mention that because an axon always travels away from the nerve cell body. So if this were the sensory cell up here, sensory neuron up here, the cell body would be up here, and this would be the axon to take it to an association neuron inside the central nervous system. If it's a motor neuron, then the nerve cell body of the motor neuron is inside the central nervous system, and the axon is that long part of the nerve cell that goes through a nerve and towards the next cell. Now this next cell could be a muscle cell, it could be a gland. If this were the axon of a sensory neuron, like I say, this could be another nerve cell and so on like that. What we have to recognize is that this wave of electrical change comes down here, and these things often branch, so I've represented two of them here. It's an electrical impulse change, and it gets to the end of the cell, and there's a space. These cells don't actually touch, but this impulse has to affect what goes on in this cell. So let me draw this again, only a little bit bigger, and I'll explain what it is I'm drawing as I draw it. So we have a nerve cell fiber, I'll take this one there for example, coming along, and notice I've drawn a bulge at the end there, and I'll draw that same bulge here. And here's our next cell. To put a little bit of terminology with this, the next cell has a membrane here. This membrane is the membrane of the nerve cell, of course. The space in the middle is called a synapse. And it's a space, very small space, infinitesimally small, but it's also called a synaptic gap. Important to recognize that these cells do not touch. This membrane at the beginning, at the end of the nerve cell, but the beginning of the synaptic gap is called a pre-synaptic membrane. And then it follows that the one on the other side is a postsynaptic membrane. The postsynaptic membrane is equipped with specialized proteins, of course, a specialized receptor site. So there may be a protein here, I'll draw it like a little holder of some sort, and another one here, for example. And these are receptor sites, little specializations on the membrane which, when the correct shaped molecule arrives, it can bond onto it, affix itself there, and cause some change inside that cell. The presynaptic membrane doesn't have those, only the postsynaptic membrane, and that's important. The presynaptic membrane does have features, however. It has vesicles, and these vesicles are filled with chemical substances. These chemical substances are called neurotransmitters. Now there's lots of different kinds of neurotransmitters, and I'm not going to get into their actual names in this video. That's a topic of another video, but I will mention things like dopamine, if you've heard of that one, or serotonin or GABA. Those are neurotransmitters in the brain. There's neurotransmitters in the peripheral part of the body, things like noradrenaline, not adrenaline, noradrenaline and acetylcholine. But that's the kind of things that these things are. Oftentimes they're a protein type molecule or protein derivative type molecule because cells only ever make proteins or lipids for secretion, and this wouldn't be very good if it was a lipid type molecule because this is all a watery system here. So oftentimes they're a polar molecule, and oftentimes they're clusters of amino acids, small peptides. Anyway, these vesicles of neurotransmitters are connected to the presynaptic membrane by a contractile protein. And inside this space, this synaptic gap, are calcium ions. So now the whole story is set up, and if you look at it, you can sort of see what has to happen, because neurotransmitters are the molecules that have to make the connection to the next cell. These molecules lodge themselves in these receptor sites on the postsynaptic membrane. But the question is, how does that happen? That's the story of synaptic transmission. That's this whole topic. Well, it happens this way. The sodium ions, of course, they enter. This impulse is moving in this direction. Potassium ions go up behind them. Sodium ions come in, come in, come in. It's opening the sodium gates. When this happens at the end of the axon here, potassium can get through. There's potassium gates in here. So potassium enters the end of the axon. Potassium can bond onto these contractile proteins, and what it does, quite simply, is it causes them to shorten. When they shorten, the membrane of the vesicle and the membrane of the end of the axon, this presynaptic membrane, they fuse. And if I can just take the liberty of just drawing this one having been drawn towards the membrane, it's shortened, it fuses, and the neurotransmitters leak into the space. When they leak into here, they're obviously more concentrated on this side than they are anywhere else. They begin to diffuse. And they diffuse across, and they will bond into those postsynaptic membranes receptor sites. And when they do, they're taken up here on these receptor sites. It causes a change in this membrane. And when it causes a change in this membrane, then it becomes so permeable to sodium, the sodium gates will open, sodium can enter here, and you realize that that's exactly what, I'm saying if this was a nerve cell, that's exactly what started a nerve impulse transmission before. Sodium enters, potassium goes out, changes the permeability or polarity of that membrane, and we have transmission. We have the nerve impulse transmission has made it through to the next cell. If this is a muscle cell, then we have calcium ions playing another role in here, and that's a whole different topic, but the mechanism is the same. So there you have it. This whole story is known as synaptic transmission. But before we leave it, I'd like to just mention the whole sequence of events, because it's this happens, then that happens, then that happens, and I've written all over the board here, but let me pull those pieces back out. If you can remember the structure the way it was at the beginning, then what we have, quite simply, is the impulse arrives at the end of the axon. And I should mention, this end of the axon sometimes is called a knob, sometimes it's called a bulb, sometimes if it's a muscle cell it's called a motor end plate. They can be different shapes, it doesn't matter, the story is the same regardless. But that's the first thing that happens. Calcium ions enter and cause the shortening of the proteins, contractile proteins, fusion of the vesicle, followed by exocytosis, Fourth thing that happens is diffusion of the neurotransmitters. And the fifth thing is that neurotransmitters are bonded or taken up at the receptor sites. This sequence of events could be described in 5 steps, 8 steps, depending on how detailed you want each step to become. As was true for saltatory transmission, if this system remained like that, it couldn't be used again, because the neurotransmitters are in the receptor sites. There's neurotransmitters still floating around in here. There's the last part of this topic is to restore this to its original condition so it could be used again, and that's the presence of enzymes in the synaptic gap. Enzymes, they destroy the neurotransmitters. So we could actually add a number 6, because this one will take this whole system back to where it was at the beginning. There's more pieces to add to this, but this synaptic transmission story ends with this topic. The kinds of things to add to it is what kinds of things are these neurotransmitters when they arrive at this thing? What's the impact on here? Because sometimes this neurotransmitter is making this cell more active, so we have an excitatory transmitter. Sometimes it's making it less active, we have an inhibitory transmitter. So there's more to this topic, and we'll get into that when we deal with the autonomic nervous system. Thank you very much.